Kaushik, I'm audible. Kaushik?
हाँ हेलो।
Okay.
Yes, sir.
சார் ஒரு 10 நிமிஷம் வந்துடுறா, வெயிட் பண்றோம். கொஞ்சம் கஷ்டமா first class எங்க அப்பார்ட்மெண்ட்ல ஒரு எங்க அப்பார்ட்மெண்ட் தான் இப்ப அட்மிஷன் போறேன் வரான் உங்க அப்பார்ட்மெண்ட் வரான் எங்க அப்பார்ட்மெண்ட் தான் இருக்கேன்
சொல்லுடா.
Only you. Just laptop. Oh, where are this? One first. May I know who is there online? Kaushik?
Sir, Kaushik, sir. Kaushik.
Yeah, Kaushik. Yes, Kaushik. Sorry for the delay. Had some program in the college. Governor has come, so it got delayed. Yes.
Okay.
So we will continue. Yes. Like I think we should start with vegetative propagation. Okay. I think can we mute there? Muted. It's echoing later. Other just muted. Okay. Yeah. Just call. Cell copies. Yeah. See, what is DNA copying mechanism? Yeah, you want that only, right? Cell copy or DNA copy? Yeah, cell can divide. Only the DNA which is inside divides. If a DNA inside the cell is not dividing, the cell cannot divide. Okay? Can a cell without a nucleus can divide? No. Okay? Only the cell must have DNA, and that DNA should divide, then only the cell can divide. Okay? So without DNA division, cell division is not at all possible. Can you give me an example of enucleated cell, DNA less or nucleus less cell? In our body, there are many cells, right? Do you think all the cells are having nucleus? Can a living cell exist without a nucleus? If you know, just give me an example. Is it possible for a cell to live without nucleus? No. Okay. See, our mature red blood cell. Our red blood cell. What is the function of red blood cell? Exchange of gases. Actually, it is not having a nucleus. There is no DNA, there is no nucleus in the cell. Okay, very few, especially the red blood cell, can survive without nucleus. No other cells. Okay, fine. Except red blood cells. So, can it divide them? It can survive without nucleus. Can it divide without nucleus? It cannot divide. Most of the cell, even with nucleus, also cannot divide. Because they are, where is your assignment? I gave you an imposition. No, you avoided. So tomorrow, 20 times. Okay, you forgot, so 20 times. Okay? You get my point? Only if the DNA divides, the cell can divide. If the DNA is not able to divide, the cell cannot divide. If a cell has DNA but no nucleus. Yeah, they are prokaryotic cell. Yes. by binary fission. Okay, you can't, you don't have such condition. Enucleated. Yes, what in assisted reproductive technology, in cloning and all, it is possible. They take Excel, how cloned animals are produced. Udder cell. They took the udder cell and obtained the nucleus alone. And then Excel. Excel is having only single set of chromosome. Okay, sheep egg cell. They remove the nucleus. Now the sheep egg cell is not having any nucleus. Okay, and then they introduced udder cell, means somatic cell, body cell with 2N into egg cell. They just transferred. That method is actually called what? SCNT. What is SCNT? Note it down and read it at home and tell me tomorrow. That's a technique which made the cloned animal Dolly. You know the technique cloning. Cloning. Have you heard about it? Cloning. Sheep, a Dolly was created. You're new to that? So, cloning. How animals can reproduce? How animals can reproduce sexually? We are discussing about the methods of reproduction. How animals can reproduce sexually? Okay, most of the animals can reproduce sexually only. Cloning, for how goats can reproduce. Weetla odd every kutti podum by sexual reproduction. But cloning created an animal called Dolly. Sheep, odd. Dolly is the sheep. Okay, they created it by asexual method. The technique is called somatic cell nuclear transfer. Okay, what technique it is? Somatic cell nuclear transfer, cloning. So, sexual reproduction involves what? Sexual reproduction involves what? Fusion of male and female gametes. When you can call something as sexual reproduction, there is involvement and fusion of two opposite gametes, male and female gametes. Correct? Then only it is called sexual reproduction. Okay, here no fusion of male and female gametes. What happens if male and female gametes are fused? Sexual reproduction. Gametes, zygote forms. Gamete fusion results in zygote formation. So 1N, 1N. Gametes are 1N means what? 1N. Gametes are 1N. N represents chromosome number. So, Appa, what is the chromosome number here in humans? 23. So, sperm cell brings 23 chromosome and egg cell also haploid cell. 23 chromosome. What is our chromosome number in humans? 46. Can you understand? It is 46. So, sperm cell and egg cell are required to make a zygote. This process is called what? Fertilization. Sperm cell appa, egg cell amma fuses by fertilization process. What forms? Zygote. Okay, then only it divides and develops into Hari, Rohit, me. That you are that zygote, fertilized in your amma's body. You are that zygote. I am that zygote. Okay, but here no such fusion from sperm cell involved. In this Dolly, no sperm cell is involved. They took somatic cell. Somatic cell means what? Somatic cell means what? Body cell. What are the two types of cells present in our body? Somatic cell and reproductive cell. What is the reproductive cell you have? A reproductive cell. Only your sperm cell is reproductive cell. All other cells are your body cells, somatic cells. Okay, only your sperm cell is reproductive cell. Can you understand? So, for you to have your baby, what you should have? Eh? For you to have your baby, only through your sperm cell you can have your baby, not through any of your cell. Correct? Only your sperm cell can fertilize egg cell. Can your skin cell fertilize your egg cell? No. Actually, we are sexually reproducing organism. Okay? But here what they took somatic cell, udder cell. பால் கறக்கிறதுக்கு மடி இருக்கு இல்லையா, that is called udder cell. அந்த அடர்ல இருந்து கோட்டோட அடர்ல இருந்து ஒரு செல் எடுக்கிறாங்க. அதுல இருந்து என்ன எடுக்கிறாங்க? நியூக்ளியஸ். That is egg cell எடுக்கிறாங்க. From the biological mother, egg cell is taken. இந்த egg cell ல இருந்து இருக்கிற நியூக்ளியஸை ரிமூவ் பண்ணிடுறாங்க. That is called enucleation. They remove nucleus. Now, somatic cell nucleus is added to egg cell nucleus. It looks little confusing. Test tube baby is not like that. Test tube baby means upper sperm cell, a man's sperm cell is taken. Women egg cell is taken. Both are used in the lab. That is called test tube baby. Only fertilization occurs in the lab. Then in day 1, day 2, day 3, they transfer that zygote embryo into mother, biological mother or surrogate mother. The baby will not grow in test tube. Only the fertilization process occur in the test tube. Okay, so that's different. That is assisted reproductive technology. Asexual, that is not asexual, that is sexual. Okay, only cloning is asexual, but human cloning is banned. You cannot do human cloning, but animal cloning, lot of things we have done. Cloning, asexually reproducing animal. Asexual reproduction of sexually reproducing organism. You are a sexually reproducing organism. By cloning, we can make another rocket. But human cloning is banned, but all animals can be cloned. So, let me conclude. Listen. Humans and animals reproduce sexually. This technique is somatic cell nuclear transfer. It's a asexual reproduction method of animals. Okay, somatic cell nuclear transfer. That's why I asked you to note down and study a little more against it. Okay, anyway, maybe in your higher classes and all, you get some clarification in this regard. Okay, so why I just, you may look, why sir is talking about all these things? Actually, this chapter is about what? How do organisms reproduce? Okay, cloning is a method of reproduction, advanced method of reproduction. Asexually also in lab you can make animals. Okay, artificially. Okay, good. Kaushik, you able to understand?
Yes, sir.
Yeah. Okay, please note down about the somatic cell nuclear transfer technique and please read it. Okay.
Okay.
Actually, in your higher classes and all, even actually not even in your +2, only in your higher classes like colleges, you read about these techniques. Genetic engineering, when you study there, you study about these techniques. Animal biotechnology, you study about these techniques. Okay, fine, good.
Okay.
Fine. Now it is vegetative propagation. So what is vegetative propagation? Plants, this is seen in plants, develop from the body parts like, okay, many plants develop into new plant under appropriate complete conditions like root, stem, leaf. Okay, plant in which parts like root, stem, leaves develop into new plant under appropriate condition. This method is called what? Vegetative propagation. Okay, vegetative propagation. So, which part it develops into the new organism? They are called what? Vegetative propagules. What are vegetative propagules? The part from which the plant develop into new organism. I will give you some simple examples. Have you seen potato? So, especially in this moist condition, you just keep a potato unused for few weeks. Okay, you can see the potato plant grows. Okay, உருளைக்கிழங்கு எடுங்க, சும்மா ரூம்ல ஒரு மூலையில வச்சிருங்க. நீங்க ஒன்றும் பண்ண வேணாம், சும்மா ஒரு ரூம்ல ஒரு மூலையில வச்சிருங்க, யூஸ் பண்ணாதீங்க, அப்படியே விட்டுருங்க ஒரு few weeksக்கு. The potato, that is simply starch, right? Maybe, but you can see as before it's rotting, you get the growth. Okay, you can see the new plant comes out, emerges out of that. Okay, the part from which the potato develop into new organism is called what? Vegetative propagule. Actually, especially in potato, it is called what? Ice. EY. Ice. Potato. Ice. Can you understand? Yeah, kutti kutti, like that ginger. Okay, in ginger also you can see nodes and internodes. Okay, what you can see? Nodes, internodes. Inji la pathina anga nodes irukku illaya, just leave the inji for few weeks untouched. Okay, you can see the growth of the plant, new plant arises from that. Can you understand? Like that. Have you seen onion? Onion. Okay, just leave the onion for few weeks, few days. லேசா நினைச்சிட்டு வச்சிருங்க ஆனியனை. கீழே பாத்தீங்கன்னா அப்படியே வளர்ந்து வரும், செடி மாதிரி வளர்ந்து வரும். ஓகே, the part உள்ள இருக்கிற பார்ட்ல இருந்து தான் ஆனியன் grows actually. From that onion part, it has the potency to develop into new organism. அதுல என்ன சொல்லுவாங்கன்னா actually bulb அப்படின்னு சொல்லுவாங்க. என்ன சொல்லுவாங்க? bulb. Okay, most of the plants. Okay, not all the plants, but most of the plants can grow. Onion, it is called what? Bulb. Can you understand? So like that. So they have the natural process. Naturally, all this can grow. Some plants, like you can see in the next page, you can see buds, bryophyllum. This is the plant. Okay, from the leaf, buds comes. Actually, this is the leaf. You can see buds. Okay, leaf, and then buds comes, and then a new plant grows. Can you give me example of vegetative propagules? Ginger nodes, bulb, potato, ice, leaf, bryophyllum, leaf buds. Bryophyllum, leaf buds. So they are called actually what? Vegetative propagules. Okay, from the bodily part, the organism can. There is no sexual reproduction involved. There is no fusion. There is no fertilization. Just vegetative propagules arise, and then the plant, new plant grows from that. வாழைமரம்லாம் சொல்லணும் இல்லையா, வாழடி வாழையா. Similarly like that. From the bodily, ஒரு வாழைக்கண்ணு வச்சுட்டீங்கன்னா போதும், ஆட்டோமேட்டிக்கா என்ன ஆகும்? பக்கத்துல சின்ன சின்ன கண்ணுங்க வர ஆரம்பிச்சிடும். Can you understand? So that is called what? Vegetative propagation. Okay, we can also do that. See, this is natural vegetative propagation. Something called artificial vegetative reproduction also. You know artificial methods? I think in your lower class you would have studied. What are the artificial methods you can see? Grafting. Okay, cutting. Okay, mounting. Have you heard about this? Grafting. What is called grafting? See, for example, yes. Take one branch from one plant. Take another branch from another plant. For example, this is a Chembarathi, hibiscus plant. Okay, the flowers are pink in color. Okay, and one Chembarathi plant, hibiscus plant, the flowers are multi-whorl. Okay, whorl, WHORL, multi-whorl, many layer of petals. Okay, but this is red. Okay, pink flower, single whorl. Red flower, multi-whorl. Okay, now you cross it. Take a branch from here, one plant, take a branch from here. You just tie it here. Now a new plant arises, which may have what? Multi-whorl pink or single whorl red. Okay, either this or both, anything depends, can come. So that is called what? Grafting. For example, autograft. What is called autograft? In humans, we can be doing this autograft. Autograft, not autograph, autograft. Autograft. Autograft means what? See, like I met with an accident. My face get injured. I need a skin to replace this, the lost skin. So what people will do, have you heard about it? They cut a skin from thigh and put it there. This is called what? One part of, in the same auto cell transplantation. I transfer a piece of tissue from here and here to here. This is called autograft. Graft means a piece of tissue. Okay, here grafting is two tissues, two plants are fused together and a new plant is developed from that. From the bodily part, two plants are fused and it is developed. That method is called what? Grafting. Can you see? Cutting. For example, what is cutting? In your house, in Hari's house, there is an in the Murunga Mara irukku, drumstick tree irukku. Na enna pandren? Come on the unga Murunga Mara, like peri Murunga kaatharudhu. Na enna pannalam? Hari, give me one kutti branch, Hari. Okay, from the stem, give me one small piece. Cut panni eduthu vanduruvan. I'll put it in my home garden.
Sir, what they do with the branch?
Only branch. Inside that, it has cambium tissue. Branch will have what? Cambium tissue. That will have regrowth potentials. Can you understand? That branch will have regrowth potential. That method is called what? Cutting. Can you understand? Something called mounting or layering. Layering. They call it as what? Layering. See, a plant grows. The branch is tall. This branch gets affected. Okay, main branch. What they do, listen, this is the soil. This is the soil. They just bend this branch and put it like this and cover it with soil and nutrients. Can you see? The main part is affected, but the side branch is only there. What do? They just bend it. Okay, put it in the soil, cover it with the soil in the land, supply manures and fertilizers, the plant will grow now. Like a new root emerges from branch. What is going to emerge? New root is going to emerge from the plant, so new plant is going to come. Like that, the creepers are growing. How creepers are growing? Creepers, pathirkingla, darbushni alla. Stem, develop root, grow. Stem, develop root and grow. Can you understand? So that mounting, layering, these are all what methods actually? They are all what actually? Cutting, layering, mounting. Basically, they are what? Methods of vegetative reproduction. Can you understand? No seeds are involved here. Okay, look at that. What I'm telling. Layering, grafting. Can you understand? In agriculture, all these simple processes are done. They are not ஒரு international technology. நம்ம ஊர்ல தாத்தா பாட்டி பண்ற ஒரு simple techniques தான். Cutting, grafting, layering and all. Actually, vegetative propagation. All our tata party will do. Can you understand? Vegetative propagation. Okay, fine. But now, since I told you genetic engineering, what did I tell? Genetic engineering. What we do here? What we do here? Or in advanced technique, we do tissue culture. What we do? Tissue culture. See, what is the season for banana now? In which season you get mango? Summer. In which season you get banana? All season. How? How it is possible? Because we do tissue culture. Banana are propagated to banana plants. Chinna plants are actually propagated through what? Tissue culture. Okay. So there is one well-known scientist here in Tamil Nadu. Okay. I think his name is Dr. Bharati. Okay. Note down his name, you just search. Always learn something out of your textbook. Dr. Bharati. Okay. I think his company is located near Osur. I'm not sure. Let me check. I think Osur only. Bharati. It's a biotech company. What do they do? Plant tissue culture. Okay. What do they do? Plant tissue. Bamboo and banana are their famous products. Bamboo planting and banana planting are their famous production. Bamboo and banana. Tissue culture means what do they do? Look at that, the steps. New plants are grown by removing tissue from the living plant. Okay. A plant or a body part is taken. For example, I've seen carrot. Carrot பார்த்திருக்கீங்களா? Carrot cut பண்ணிருக்கீங்களா? Carrot cut பண்ணா உள்ள ஒரு tissue, உள்ள பேல ஒரு ring தெரியும். What is it actually? Cambium tissue. அந்த tissue பேர் என்ன சொல்லுவாங்க? Cambium. இதெல்லாம் 9th standardல இருக்கும். Tissues அப்படின்னு 9th standardல படிச்சீங்களா? Tissues. Cambium tissue. Cambium tissue படிக்கலையா? Simple tissue, complex tissue படிச்சீங்களா? Permanent tissues, அதாவது meristematic tissue, permanent tissue படிச்சீங்களா? Meristematic tissue ஓட type என்ன? What are the types of meristematic tissue?
Sir, I have apical, lateral, and
apical meristem,
intercam.
lateral meristem. What are the type of lateral meristem? Note down this and check it in your book and tell me. Core cambium. Do you remember? Cambium tissue. Okay. Basically, this cambium tissue gives the secondary growth. What is primary growth? What is secondary growth? The seed germinates, root forms, shoot forms, the plant grows. That's primary growth. What is secondary growth? If you cut the plant, well-developed plant, you cut it. Just leave the root alone. The plant regenerates. That's because of what? Not because of the seed. Do you have seed there? No seed. But how the plant grows? That's secondary growth because in between its permanent tissue, it has cambium tissue. Please refer your class 9 tissues chapter. Yeah, sugar can grow vegetative from the bodily part because it has cambium tissue. But with meristematic activity.
So then why is it only 3 times? Why is it only 3 times?
Slowly, every cell will lose its viability. For example, you have stem cells. Do you think your stem cell, why you are getting aging process? Your skin cells are having meristematic activity. Old cells, even when you walk, your cells are living out from your body. Because every day your old cells are, every day you lose 1 gram of cells. How many? 1 gram of cells, epithelial cells, you own out every day. You are losing your dead cells 1 gram per day. ஒரு நாளைக்கு 1 gram of cells அவனோட பாடியில இருந்து வெளில போயிட்டே இருக்கு செத்து. That's why, because they are regaining. New cells are replacing the old cell. Yes. Why கடிச்சிக்கிற முறுக்கு கடிக்கிறன்னு வாயை கடிச்சிக்கிற காயம் சரியாவுதா? செத்து போன செல்ஸ் போயிடுச்சு, புது செல்ஸ் வருது. அதே மாதிரி இப்ப நீ உனக்கு ஓல்டு செல்ஸ் போறதுனால எங்கேஜ்ல ஃப்ரெஷ்ஷா இருக்க, உனக்கு தெரியல. தாத்தா பாட்டிக்கு ஏன் தோல் எல்லாம் சுருங்கி போகுது, காயம் பட்டா ஏன் ஆற மாட்டேது? Because they lose the potentials actually. The cells slowly undergo aging process. உனக்கு முடி கொட்டினா வளர்ந்துரும், எனக்கு வயசாயிடுச்சு, எனக்கு கொட்டினா வளராது, கொட்டினதுதான். Look at my head. Can you understand? Aging process. Cells will undergo aging process because they loses their potentials. No, it cannot regenerate. For me, aging process. Lost hairs or lost only.
Slow. It doesn't stop, but it's slow.
Slow.
The regeneration process.
No.
It's slow.
Slowly, and then completely loses it. Yeah. That's what aging process. Slow, progressive aging process. It's called what? Progressive aging process. Even if your bone gets fractured, it will regrow, remodel itself. But for aged people, even the bone cannot regrow actually. They have to carry like a lot of means they do not regrow. They have to put rod and all. They have to set it. No growth is seen because of the aging process. Can you understand? So like that, even it is meristematic tissue, even in plants, 1 cycle, 2 cycle, 3 cycle it grows. They are not immortal. But bacteria, unicellular organism, bacteria or what? Yeah, they are actually immortal. Yeah, bacteria. Under proper condition, they don't die. They are immortal, unicellular or immortal. Kaushik, can you understand? Bacteria under proper appropriate condition, they are immortal. Because one bacteria, by binary fission, one bacteria, that itself divided into 2. Yes, sorry.
Can we just kill them by burning?
Yeah, that's what I said. Under appropriate condition, they will not die. If you heat it, it will die. Why you milk? Every day you get your milk packet, which is called what? Pasteurized milk. Have you seen pasteurized milk? What is called pasteurization? It is heat killing. Just heat killing the bacteria. Unwanted bacteria are killed in your milk. Sometimes it contains bacteria. See, ஊர்ல பசும்பால் காலைல வாங்கினா 11 மணிக்கெல்லாம் கெட்டுடும். பசும்பால் வாங்கி வச்சீங்கன்னா 2-3 4 மணி நேரத்துல கெட்டுடும். பட் நீங்க குடிக்கிற பால் 2-3 நாளைக்கு முன்னாடி ஆவின் ஃபேக்டரிக்கு வந்தது. அவங்க என்ன பண்ணிருக்காங்க? அதை ஹீட் கில் பண்ணிருக்காங்க. பாஸ்டரைசேஷன். ஹீட் பண்ணுவாங்க, கூல் பண்ணுவாங்க, ஹீட் பண்ணுவாங்க, கூல் பண்ணுவாங்க, ஹீட் பண்ணுவாங்க, 3 times they were done. That's right. The harmful bacteria are dead there. You can kill by heating. You can kill, of course, by burning. Why hospitals லாம் போறீங்க, medical waste எல்லாம் they call it as what? Incineration. Burn it. Medical waste என்ன பண்ணுவாங்க? Burn பண்ணுவாங்க. That process is called what? Incineration.
You said heat it and cool it, right? But if we heat milk and keep it in fridge, won't it get bad?
இல்ல, ஹீட் பண்ணி கூல் பண்ணி ஹீட் பண்ணி கூல் பண்ணி ஹீட் பண்ணி கூல் பண்ணி என்ன பண்ணிடுவாங்க? அதுல இருக்கிற டெட் பாக்டீரியாவை கொண்டுடுவாங்க. அந்த பாக்டீரியாக்கு நேச்சுரலி என்ன ப்ராப்பர்ட்டி இருக்கும்? மில்க கர்டா மாத்த, மீன்ஸ் ஸ்பாயில் பண்ற. நாட் ஈவன் மில்க் டு கர்ட், இட் ஜஸ்ட் ஸ்பாயில்ஸ் த மில்க். அதனால அதை மட்டும் நம்ம அழிச்சிட்டோம். பட் அதுல என்ன இருக்கும்? ஆக்சுவலா லாக்டோபேசிலஸ்ல இருக்கும். நாட் ஆல் த பாக்டீரியா ஆர் ரிமூவ், பட் தே கேன் சஸ்டைன். So they will be there. அதனால நீங்க மில்க் கர்டா மாத்திக்கலாம். பட் ஈவன் லாக்டோபேசிலஸ் இல்லைனாலும் பரவாயில்லை. நீங்க என்ன பண்ணலாம்? Sometimes see, recently, like ஒரு few months back also, I got a milk packet called Avin Delight. Kaushik, you know that? Avin Delight. பால் பாக்கெட் வாங்கினா எங்க வைப்பீங்க? ஃப்ரிட்ஜ்ல வைப்பீங்க. ஓகேயா? பட் இந்த Avin Delight 1 மாசத்துக்கு for 30 days. For 30 days சும்மா இப்படி நீங்க பொட்டலம் முறுக்கு வாங்கினா இப்படி வைக்கிற டப்பால வைக்கிற மாதிரி வச்சுக்கலாம். I used that Avin milk. Very good. Okay, but no, இப்ப கிடைக்கிறது இல்லை, என்னவோ தெரியல. But it was there in the market. You just check. Avin Delight. என்ன சொல்லுவாங்க? Avin Delight அப்படின்னு சொல்லுவாங்க. LIG.
So you keep it out, right?
ஆமா. Not in fridge. It is not refrigerated. Packet, milk packet. Ama. Boiled, filtered. All the pathogens with all the bacteria removed. No bacteria attack. Just protein, ions, minerals, water is there. No, not even a single bacteria is there. இப்போ நம்ம இந்த வாட்டர் பிடிச்சி வச்சிருங்க ஒரு பாட்டில்ல, ஒரு கிளீன் பாட்டில்ல. டேக் திஸ் ஃபில்டர்ட் வாட்டர், ஒரு டேப் வாட்டர். ஒரு 1 month கழிச்சு பாருங்க, this water is pure, almost pure. But நீங்க பிடிச்ச டேப் வாட்டர்ல புழு வந்துட்டு இருக்கும். That is not fit. There it has, you have bacteria, but here you don't have bacteria. If bacteria is there, it may change the quality. Okay, so in Almond Delight milk வெளியில வச்சாக்கும் கூட வரவே வராது. Can you understand why we went there? Always hurry in a okay, anyway, good. Anyway, just all these things actually you should know basically. Okay, so back pasteurization, no issues anyway. So pasteurization, the bacteria, okay, like basic common man science only. All these things which we discussed are the common man science. Okay, fine. So vegetative propagation. Okay, what is vegetative propagation? Plants reproduce by bodily parts like stem, root, leaves. Okay, and then asexual method of reproduction. And the part from which the plant grows, they are called what? Vegetative propagules. Examples of vegetative propagules: ice, potato, onion bulb, bryophyllum leaf. Okay, ginger nodes and internodes. Okay, fine. And now plant tissue culture, like they take a plant, sterilize it, cambium tissue, please understand that. Okay, that cambium tissue will have meristematic activity. That tissue is sterilized. Plant tissue culture medium, okay, it is called MS medium. Please note down, the medium is called what? Murashik and Skook medium. And just read it, Murashik and Skook, just read it from the net, the spelling. MS medium, the plant tissue culture medium. Okay, search in the net. It is a scientific, ஒரு சயின்டிஸ்டோட நேம் அது, அதனால அப்படி வரும். அப்புறமா நெட்ல பாத்துக்கோங்க. Okay, listen, take a piece of plant tissue that is called explant. Okay, plant leaf, for example, leafல இருந்து ஒரு பீஸ் எடுங்க, that is called explant. Sterilize it. Sterilization என்ன? Surface sterilization. Clean. எத்தனை நாள்ல போட்டு வைப் பண்ணுங்க, அதுல இருக்கிற bacteria or whatever contaminating organisms will go off. Okay, and then sterilize பண்ணிட்டு that MS medium போட்டு லேப்ல வச்சீங்கன்னு சொன்னா, lab what forms? Callus forms. Callus, what is called callus? Unorganized mass of cells. அதாவது cells cambium tissue ல இருந்து சின்னதா நீங்க cambium வைப்பீங்க, that develop into what? ஒரு பெரிய டிஷ்யூவா மாறும். And then this tissue becomes what? A plant. Root varum, aprom branches varum, aprom the plant emerges. This technique is called what? Plant tissue culture. Tissue culture or plant tissue culture. So in lab also you can cultivate the plant. In lab also you can cultivate the plant. Okay, for this you don't have to wait for the season kaga you don't have to wait. Can you understand? So all season, every day you can do it. Multiple plant. Artificial, but no other option. Okay, otherwise, நீ தடிக்க விழுந்தா வாழைப்பழம் இருக்கும். Everywhere you can see banana. All type of banana. If you go to one shop, specifically if it is a banana, if you want பச்சை வாழைப்பழம், you can get. If you want that yellow color பூவம்பழம், you can get. கற்பூரவல்லி, you can get it. செவ்வாழை, you can get. Can you understand? Almost 4 or 5 type of banana in one shop you can get everywhere. Can you understand? So that's because of what? Only because of the plant tissue culture. Vegetative propagation. That's because of what? Vegetative propagation. Yeah. See, not all the plants can grow in lab conditions. For example, can you do this for apple? Apple. See, what did they do? They just make a simple plant. குட்டி பிளான்ட் they make பா, அந்த சின்ன செடியை கொடுத்துருவாங்க அக்ரிகல்ச்சர் ஃபார்மர்ஸ் கிட்ட. ஃபார்மர் எடுத்துட்டு போயிட்டு அவன் நிலத்துல போடுவான், அது வளரும். அந்த இனிமே இந்த சின்ன பிளான்ட்டா வர்றதுக்கு தான் உனக்கு லேப் தேவை. அதை பெருசா தோட்டமா வளர்க்கறது உனக்கு என்ன தேவை? வயல் காடு தேவை. அது பனானா நம்ம ஊர்ல வளர்ந்துரும், அதனால பனானா செடி கொடுத்தா நீ வளர்த்துருவ. உனக்கு ஆப்பிள் செடியை கொடுத்தா உன்னால வளர்க்க முடியுமா? ஏன்னா இந்த கண்டிஷன்ஸ், என்விரான்மெண்டல் கண்டிஷன்ஸ். அதுக்காக லேப்ல உட்காந்து ஒரு ஆப்பிள் ட்ரீ ஒருத்தன் வளர்த்துட்டு இருக்க முடியுமா? நம்ம கிட்ட லேப் கிடையாது. சின்ன செடி மட்டும் தான் பண்ண முடியும், மரம் பண்ண முடியாது. அவ்வளவு பெரிய ஃபார்மும் நம்ம கிட்ட கிடையாது, அவ்வளவு பெரிய கண்டிஷன்ஸும் நம்மளால கொடுக்க முடியாது. பண்ணி கொட்டணும், நல்லா சில்லுன்னு இருக்கணும் கிளைமேட். அப்பதான் எங்க ஆப்பிள் கிடைக்குது, வெரி கோல்டு ஏரியா, ஊட்டி காஷ்மீர்ல தான். காஷ்மீர், ஹிமாலயாஸ், அங்கதான் நல்லா கிடைக்குது. ஏன்னா அந்த கண்டிஷன்ஸ். கேன் யூ மேக் இட்? வெளியில வயல் காட்டுல பண்ண முடியுமா? முடியாது. ஸோ யூ கேனாட் ப்ரொவைட் சச் கைண்ட் ஆஃப் கண்டிஷன்ஸ். ஸோ தட்ஸ் வை நாட் ஆல் த யூ கேனாட் கெட். நீ கேட்கிற லிச்சி கிடைக்காது. ஓகே, பட் பனானா என்றால் யூ கேன் கெட். கேன் யூ அண்டர்ஸ்டாண்ட்? நம்ம ஊர்ல வர, அதனால எல்லா சுகர் கேன், மெனி ரைஸ், we need rice, lot of rice varieties are grown here. நீ கடைக்கு போ, சிவாஜி இப்போ என்ன வெரைட்டி ஆஃப் அரிசி கேட்டாலும் கிடைக்குது. Can you understand? So that's because of what? Tissue culture and genetic engineering. So vegetative propagation and artificial in the plants எல்லாம் இப்படி பண்றதுனால தான். If you know about all these techniques, ஏன் இதெல்லாம் நீங்க படிக்கிறீங்க? Why I should tell about all these things? Lot of scope we have. Biology na enada scope irukku. If you learn just one technique, the vegetative propagation technique, banana sampling enakku panna theriyum, you are a multimillionaire here. Can you understand? You can make many plants, supply ore vishayatha pannite irunga, supply to every farmer. You are a businessman, entrepreneur there. Can you understand? Yeah, many. That's what the crop plants. We get varieties of rice plant. We get varieties of wheat, corn, oil producing plants, paruthi chedi. Yeah, not just banana. Many plants are cultivated like that. If you only rely on natural organic thing, very difficult to get. Okay, because conditions are changing. No, good. You get good quantity, means good quality of materials, but not quality. Maybe only the plants are produced there, but where do you grow it? You are not injecting any medicine and all. Where it grows actually? In the nature da anga nee potrudha porthu da. It is not depending upon the company. Pesticides, if you do it properly, you will get good thing only. Of course, if you look for more quantity, you have to slightly compromise your quality. Okay, so because in those days rice plant takes 6 months to grow. கேள்விப்பட்டிருக்கீங்களா? 6 மாசம் ஆகும். ஆடி மாசம், அதாவது ஆடிப்பட்டம் தேடி விதைன்னு சொல்லுவாங்க தமிழ்ல. ஓகே, they do it in the month of ஜூலை ஆகஸ்ட்ல தான் விதைப்பாங்க. எப்ப கல்டிவேட் பண்ணுவாங்க? பொங்கல், ஜனவரி. That's why we celebrated பொங்கல் there. அது வரை they have to take care of that. But இப்ப இருக்கிற காலத்துல is that possible? உட்கார்ந்திருக்க முடியாது. But that's why, who is, I told you, M.S. Swaminathan, father of green revolution, he introduced a semi-dwarf. அதாவது பிளான்ட்டே அந்த காலத்துல ரொம்ப ஹைட்டா வளருமா, அப்புறம் தான் ரைஸ் எல்லாம் ப்ரொடியூஸ் ஆகும். இவர் என்ன பண்ணிட்டாரு? செமி-டார்ஃப். நீ ஏன் அவ்வளவு தூரம் வளர போற? மனுஷனே வளர்றது இல்லை, நீ வளர்ந்து என்ன பண்ண போற? உனக்கு எதுக்கு 3 மாசம் வளர்றதுக்கு? ஜெனட்டிக்கலி மாடிஃபை பண்ணி நீ பாதி வளர்ந்தா போதும், உடனே எனக்கு அரிசியை விதைய கொடு. So 6 months become 3 months. Can you understand? அப்ப நீ இப்ப என்ன பண்ணலாம்? 2 டைம் அறுவடை பண்ணிடலாம். Can you understand? நவம்பர்ல போடுறான், இப்ப ஜனவரியில எடுத்துறோம். அதை விட கம்மியாச்சு 60 days. Can you understand? Not even 90 days. Now in 60 days you get, from the seed you get a whole plant, lot of varieties. Can you understand? So that is assisted reproduction, means in a vegetative propagation, plant tissue culture. Can you understand? So lot of genetic engineering, of course. Okay, anyway, so but just one more topic we just see and finish. Okay, spore formation. See, listen, all asexual reproduction method will finish. Look at that. After vegetative propagation, spore formation. See, look at that. Like rhizophus, organisms like rhizophus, look at that. In their bodily part, they produce an unit. Actually, they are called what? Sporangium or sporangia. Sporangium, plural is sporangia. In their body part, they produce a particular unit. What are they? They are called sporangia. Inside that, can you see kuti dots there? Inside that, can you see a kuti dots there? They are called what? Spores. Okay. So the plant develops and they releases the burst, the sporangium bursts and releases the spores into the air current or water current. They reaches different part, maybe by wind or whatever, and wherever they're landing here, the whole plant develops. They're landing here, the new plant develops. That method is called what? Spore formation, sporogenesis, spore formation. Okay, in simple multicellular organism, like thread-like structure, basically like they have a hyphae, whatever, and then in there they have sporangia. Can you see? Bulb-like blobs or sporangia. Okay, the model is called what? Tiny blob-monistic structure. Okay, sporangia are what? Tiny blob-monistic like structure. Can you see? ஒரு சின்ன கோடு அது மேல ஒரு பிளாக் பல்பு மாதிரி ஒரு பால் மாதிரி, that is called sporangium. அங்க இருந்து spores are produced and that spores spread. Okay, spores they can develop into what? New rhizopus individuals. Can you understand? So dry condition la spore ஆயிடும், moist condition la fungus எல்லாம் எப்போ வளரும்? Moist condition la தான். ஓகே, மழை காலத்துல பாருங்க மரத்தெல்லாம் அப்படியே மஷ்ரூம்ஸ் எல்லாம் வளர்ந்துருக்கும், தட்ஸ் a fungi. நாட் வெயில் காலத்துல வர்றது. பாரெல்லாம் பாருங்க மழை காலத்துல அப்படியே வழுக்கும், வீடு படிக்கட்டே வழுக்கும், ஏன்னா அங்க ஃபங்கை வளர்ந்துருக்கும். இந்த மாதிரி ஏன்னா எப்படி அங்க நீயா போட்ட விதை ஏற்கனவே வந்த ஃபங்கை ஸ்போர்ட்ஸை விட்டுட்டு போயிருக்கு. நீங்க ஃபேவரபிள் கண்டிஷன்ஸ் வந்தோடனே the sports regenerating, மாய்ஸ்ட் கண்டிஷன் கிடைச்ச உடனே they begin to grow. ஓகே, இப்போ ஃபார் எக்ஸாம்பிள் இந்த கீழே இந்த படிக்கட்டுல ஓரத்துலாம் பாத்தீங்கன்னா எதுவுமே இருக்காது, ட்ரையா இருக்கும். ஆக்சுவலி வாட் இஸ் தேர்? அந்த இடத்துல ஸ்போர் இருக்கும். ஈரம் பட்ட உடனே ஸ்போர்ஸ் வில் ரீஜெனரேட், grow into a fungus. Can you understand? So that method is called what? Spore formation. Okay, fine. So all these things are reproduction. Fungi reproduces by spore formation. Rhizopus is a fungi. This is a fungi. They produce the spores. The spores can make another organism wherever the spore spreads. அதனால தான் இப்போ இங்க பாருங்க dandruff இருக்கு உங்களுக்கு, ஃபங்கஸ்னால வருதுதான். ட்ரையா இருக்கும் தலை, சொரிஞ்சிட்டீங்க என்ன ஆயிடும்? இங்க இருக்கிற ஸ்போர் இங்க போயிடும், பார்த்தா இங்க dandruff வரும். ஸ்கின் இன்ஃபெக்ஷன், இங்க சொரியுறீங்க இங்க படுது, அப்புறம் இங்க சொரியுறீங்க, பாடி ஃபுல்லா ஸ்கின் இன்ஃபெக்ஷன் பரவிடும். Can you understand? Because the spores you are transferring, இங்க ஈரப்பட்ட உடனே இங்க வளரும். Can you understand? So fungi reproduces by spore formation. Bread mold எல்லாம் பார்த்திருக்கீங்களா? பிரெட் சின்னதா ஒரு இடத்துல வரும், அப்புறம் பார்த்தா ஃபுல்லா பரவிடும், ஃபுல்லா பிரெட் கெட்டு போயிடும். ஃபங்கை ஸ்போர் தான் அப்படியே பரவி வந்துடும். Especially like சில fungi are good fungi, but some fungi are toxic. They can cause us diseases. Like either you can antifungal agents, you get skin infection, antifungal, dandruff, anti-dandruff, antifungal agents are used actually. Can you understand? There are medicines, antidotes are present. Take care. Thank you. Sir, please stop it.
And you was